PhD student with SLU in Umeå at the Department of Forest Remote Sensing. And I want to talk a bit today about my PhD project. So this PhD is part of a bigger project called Landscape Breeding, a new paradigm in forest tree management. And the idea is to develop a new digitized game-changing tree breeding strategy that overcomes the limitations of conventional tree breeding. And we want to do that by working and operating in commercial forests directly and by accounting climatic and environmental variables. And it's more or less a combination of quantitative genetics and implementing remote sensing techniques into this. And the idea is to more or less speed up the breeding cycle for the Swedish tree breeders. So for the background, with climate change, we expect warmer temperatures and drier summers, and there is a need to identify resistant trees through the tree breeding program. And obtaining that data is rather resource intensive at the moment. They do this by hand, go to the field every 5 to 6 years and study that. And remote sensing could be an alternative to have a substitute to manually collect the data and do this with remote sensing and make it more cost effective, and also have an opportunity to get data more than only 5 to 6 years. So the goals for that are that I want to see if the timing of bud flush for young Norway spruce trees can be assessed through high resolution multispectral images. Just so everyone knows what bud flush is. Bud flush is when you have a twig and you have the buds there, and then in summer they swell, they break open, and then from there the new twigs, more or less, or the new shoots develop, and that's the process of bud flush. And then I also want to see if damage can be detected from these high resolution multispectral images. And then the final step will be to see if there is a relationship between bud flush damage and the genetic of the tree. So methodology-wise, I did field season in spring 2023 and 2024, did fly a multispectral high-resolution drone in 25 meters height, and also collected reference data. The second step is then to take these images, process them, and generate 3D point clouds, also mosaics and digital surface models. Then the third step is to have an image analysis where we'll extract the information for every single tree on site and then go into statistical modeling. So the data collection, my study sites are in southern Sweden in Småland. As mentioned before, we did field season in 2023 and 2024. These study sites are progenitor trials from Skogfors. So these trees were planted in 2018. They're around 1.5 meters tall, and we know the genetic origin and know the whole history for every tree on site. Then for the reference data, the one I want to focus today on is bud flush. So this bud flush scale according to Crutch can be assessed to that scale you can see here, starting with stage 1 and 2, where the buds start to swell and then slightly open at 3 and 4, and then the new shoots develop and lignify into the stage of 8. We also did assess the vitality of the whole tree and the damage of the new shoots. The study designs look something like this. So they are nicely organized in rows and blocks. So we decided to sample for each of these blocks, sample in row 3 and in row 8, and there sample every 4th, 8th, and 12th tree to get an even spread on the study site for the modeling later, rather than to focus more on the genetic background. For image processing, the most crucial step here was to align the flights to each other so that every tree top is more or less stacked on top of each other. So when I later do the pixel value extraction, they have perfect alignment and I don't have to worry about that. And then from there, we generated digital surface models and also mosaics, and we did use arches of metashape for that. I want to show you this time series to make it a bit more clearer what I'm looking at. So this is a snip from an orthomosaic. That's the same tree starting at the end of April and then going through the vegetation season until the end of July. So what you can see here, we start here, you have some shadows. The tree is still more or less in a dormant state. Then it progresses to here. Here the bud flush really kicked in. You can see some smaller twigs. And then if you compare this tree to this tree, it looks more or less like a complete different tree. What you can also see is that we have some shadows here and there and different effects of illumination. And that's why we did radiometric correction. So we had 4 spectral targets on site for every flight, and then we did calculate a regression on these spectral targets. And here we used the first week as a reference, then applied the regression results on all of the orthomosaics, and then I got spectrally corrected orthomosaics. Then I needed to identify the trees on the study site. So for my reference trees, I just did measure the GPS positions for them. And then for the other trees, I used a maximum detection filter on the digital surface model. And when that is done, then it looks something like this. So on top of the measured GPS positions, you can also see that it did identify the single trees there. But what you can also see if you zoom in, that it did not only detect single trees, but I also detected some stumps that were still on the side or rocks. And there's still this issue with like slight shadows in the crown. So what I did do in the next step is I applied some thresholds to more or less obtain purer pixels in the study site and get rid of these stumps and rocks. And then from there, the idea is to use the maximum of the tree as an anchor and then anchor a 30 centimeter radius around the local maxima and extract the mean, minimum, and maximum pixel value for that local maxima, so for each tree. What is to be done then is I need to use the reference trees to create a model that works as a function of the bud flush. So the pixel values work as a function for the bud flush. And then from there, I can generate the bud flush for the non-reference trees and also get the stages for each single tree. And then I will also repeat that for the damage and the vitality. And then the idea at the end is that I will have available for each tree the timing of the bud flush, the damage, the vitality, and I have the genetic background. I will then do a analysis where I want to account for the spatial variations on site. So I want to look at the soil moisture, slope, edge effects, and then sort of put that into relationship to these variables and analyze them. And then that data will get fed into the other or to one of the other PhDs working in the project, and she will then do a quantitative genetic analysis with this data. Unfortunately, I'm at the modeling stage at the moment, and it's not quite there yet to show you, but there will be a paper out, I think, in autumn. And with that, that's it for my presentation. Thanks for listening, and yeah, I'm happy to answer any questions.